Hi, everyone. Welcome to our wrap up video, our summary video of the central dogma of biology, which is also known as gene expression. And so, what I want to do here is really recap what was going on with gene expression. And this is called the central dogma of biology because really this is what biology boils down to, how DNA serves as our genetic code. And what that actually means is it is the instruction manual for how the cell needs to build the proteins that are required for it to survive. Okay. And so, with the central dogma, aka gene expression, we are looking at, I'm going to write this vertically to align with this image. DNA is used to make RNA, and that RNA is used to make proteins. And I've already annotated this image here. The process of using DNA to make RNA is called transcription. And the process of using that RNA to make proteins is called translation. Now, I want to take a second here to try to help you remember these two terms because they sound really similar. On an exam, it's easy to get them confused. And so I want you to look at transcription as a word and then translation as a word. So I would be willing to bet a lot of people are at least familiar with the term translation, but transcription, maybe not. So, with transcription, we can transcribe things. Okay, you might have heard the words transcribe before. So, I'm just going to go ahead and give you an example. When I was working on my master's in education, I would interview teachers because I was trying to learn about them, how they implemented things in their classroom, challenges they faced, motivations, things like that. And I would go and I would interview them. And in my case, I interviewed them in English. But then I had to go home and type up that interview because if I wanted to analyze the data, it was going to be easier to do just reading it and marking up a piece of paper. And similarly, if I was going to go publish this information, I need to have it in typed format. So what I would do is I listened to my English words, right, the interview, and I sat there and I typed it in English. So in this case, the language was the same, but the format was different. And that's similar to what's happening in the process of transcription. Both DNA, which is what we're starting with, and RNA, what's being made, are both nucleic acids. Okay, remember the Na here stands for nucleic acid. And so we're speaking the same language here when we're dealing with DNA and RNA, but they're not the same thing, right? Two different names mean they're two different things. And so if you recall from the transcription video, DNA is made up of nucleotides that include a phosphate, a sugar, deoxyribose, and then one of four nitrogenous bases: adenine, thymine, cytosine, and guanine. RNA is also a nucleic acid, so it's also made up of nucleotides, but they look a little different. They also have a phosphate group. They also have a sugar, but this one is ribose. And there's also one of four nitrogenous bases: adenine, uracil, guanine, and cytosine. So DNA and RNA are not the exact same things. There's a lot of similarities between their nucleotides, but there's a few differences, but those differences matter. So when we're doing transcription, think of us as staying in the same language, but we're just writing it in a different format. Okay, so A, T's, G's, and C's for DNA, A, U, G, and C. Okay, no thymine in RNA. Then when we're going from RNA and or using that RNA to make proteins, now we're translating. We're going from one language to another. the nucleotides that make up RNA are very different from the amino acids that make up a protein. So now we're changing languages and we need a translation device or a translator to help us. That's where that codon wheel came into play. Now, some people use a codon table, but it's really the same thing. It lists out all the different codons found on messenger RNA, and it tells you what amino acid it codes for. Okay. And so that's kind of how you can keep those two terms separate, because it's going to be important on exams that when you hear the word or read the word transcription, you go, aha, DNA to RNA. When you hear translation, you need to think RNA to make proteins. Okay. And so what's happening here, y'all? Let's go back to this picture, and I'm going to erase a few things. Oops, if I could pick the right button. Here we go. Just because I don't want this picture to get too busy. All right, so let's go ahead, and we're going to use green here. So if we're starting with DNA, remember that DNA is double-stranded. So we see our two strands of nucleotides. And during transcription, that DNA is going to be used to make RNA. Remember that our RNA is always going to be 5' to 3'. Okay, always. And it's single stranded, which is what you see here, that one single, in this case, yellow strand. And we used our double stranded DNA to make it. So let's go back and review our features of DNA. So we know that DNA is double stranded. So we have the blue strand and the red strand here. We know that those strands are anti-parallel, meaning they run in opposite directions, and they're complementary. So we can see the complementary nature right away, right? Adenine pairs with thymine, and then thymine pairs with adenine, right? So we see guanine pairing with cytosine. So we can see the base pairing right there with the bases. But it's not showing us the anti-parallel nature. So we know that since mRNA is made 5' to 3', the strand used to make it has to be anti-parallel. So remember that the template strand of DNA is always going to run 3' to 5'. So that means this strand ran 5 to 3. And if you need a review on this, there is a video that talks about the structure of DNA, where I really emphasize the anti-parallel and complementary nature of these strands. All right, so during transcription, the 3' to 5' DNA strand was used as a template to build our messenger RNA. Okay, so that was made 5' to 3'. And you'll notice we have our base pairing, right? We're going to have thymine on the template DNA strand. Thymine pairs with adenine. Now we have adenine on the DNA strand, but remember, in RNA, it pairs with uracil. There is no thymine in RNA. So then we look at the next base. We have cytosine here on the DNA, which means we're going to have its base pair, guanine, on the RNA. Okay. And also notice how the nucleotides are written on the messenger RNA. Notice it's in sets of 3. Can you remember what those sets of 3 are called? A codon. Okay, so the AUG is always our start codon, and then you just number the rest of them until you hit the last one, which is called the stop codon. And so then during translation, the messenger RNA is going to serve as the directions. And so the start codon is AUG. That tells the cell, hey, I need methionine as my first amino acid. The little tRNAs, one of them is going to go out and grab that methionine and deliver it to the ribosome. So remember, y'all, we need to be keeping in mind our key players. Which enzymes or molecules are responsible for this process happening? With transcription, an enzyme called RNA polymerase was building that RNA molecule. For translation, our key players are the messenger RNA and transfer RNA, and the ribosome. And remember, the ribosome has a small subunit, a large subunit, and ribosomal RNA. Okay. And so, one last thing that I want to remind you of here is that during transcription, all types of RNA are made: messenger RNA, transfer RNA, and ribosomal RNA. Okay, but in this picture, it's just showing you the mRNA initially because that's the directions used to make the proteins. Okay, all right, y'all, that's it for now when it comes to gene expression and the central dogma. So please let me know if you have any questions. Bye, y'all.